All right, we're going to finish up our look at human heredity and continuing to look at disorders that we find in humans. And what we looked at previously are the result of mutations in genes, so gene mutations. But if you'll recall, there are also chromosomal mutations. And so you might be wondering how that happens or when it happens. And it typically happens during meiosis. So you'll notice we're looking at a diagram of meiosis here. And it happens through a process known as non-disjunction. Non-disjunction means not coming apart. Disjunction means coming apart, and put a non in front of it, that means not coming apart. And that's what happens. During non-disjunction, during meiosis, the chromosomes don't separate properly. Now, this can happen during meiosis 1 or meiosis 2. And if you'll recall, during meiosis 1, it's the homologous chromosomes that separate from each other. They're still duplicated chromosomes, right? And so if this happens during meiosis 1, that means one of the daughter cells is going to end up with an extra chromosome, and the other daughter cell is going to end up missing a chromosome. And then those are going to go through meiosis 2 and produce daughter cells, each one producing 2 daughter cells. So of the 4 cells that end up at the end of meiosis, if there is non-disjunction in meiosis 1, 2 of the cells are going to end up with extra chromosomes, and 2 of the cells are going to end up missing chromosomes. And we call the wrong number of chromosomes aneuploidy. So aneuploidy is a general term just referring to the wrong number of chromosomes, whether it be too many chromosomes or too few chromosomes. Now, of course, there are more specific terms referring to each of those conditions. And we'll get to those in a minute. But first, let's consider what would happen if non-disjunction happens during meiosis 2. The reason we're looking at only a diagram for meiosis 1 here is because that's the more serious condition. In other words, all 4 gametes are affected if non-disjunction takes place during meiosis 1. If it takes place during meiosis 2, then only 2 of the gametes are going to be affected. One will have an extra chromosome and one will be missing a chromosome, and the other 2 will be normal. So it's less severe when non-disjunction happens during meiosis 2. Or at least the likelihood that a gamete that is aneuploid will be involved in a fertilization event and produce a new individual is cut in half anyway. Now you have to remember that each chromosome can have 1,000 genes or more. And you might be thinking to yourself, well, I can understand why missing 1,000 genes would be a problem, but what's wrong with having extra genes? Well, as I mentioned previously, when we were looking at the sex chromosomes, if you have an extra chromosome, that means you have extra genes, and that means you're producing extra proteins, and that can cause problems because the amount of proteins that are synthesized in living things is very tightly controlled and has to be at a certain concentration or a certain amount. So having too many proteins or too much protein synthesis is a bad thing. Now it all depends on which chromosome it is, either missing or extra, that is going to determine what's going to happen next. But for the most part, aneuploidy and non-disjunction results in miscarriage. It's most often fatal because you can't live missing a chromosome or having an extra chromosome. You would never be born. You wouldn't develop properly. Those that survive, though, are going to be aneuploid. Or more specifically, those that are missing a chromosome are going to be monosomic. And those that have extra chromosomes are going to be trisomic. Tri referring to 3 and mono referring to 1. So monosomy is missing a homologous chromosome, and trisomy is having 1 extra homologous chromosome. And as I mentioned, there are many disorders that result from non-disjunction. It depends on which chromosome is affected. So I started out here reminding you that there are gene mutations and chromosomal mutations, and there are different types of gene mutations and different types of chromosomal mutations, and the 2 different types of chromosomal mutations are mutations of number, which is what we're looking at here, which causes aneuploidy. But remember, there's also chromosomal mutations of structure. So review, just to remind you that there are also instances where pieces of chromosomes will be missing. And again, those pieces can have hundreds or thousands of genes involved. So they're missing, if a chromosome is missing a piece, it might be missing quite a few genes. Or if it has an extra piece attached to it, that means it has too many genes, just like in the case of trisomy. So how do we know if an individual is aneuploid? How can we find out? And it's especially important to find out before the individual is even born. And if you'll recall, that's how we generated the karyotype that I showed you of our daughter. We had that done before she was born because we were concerned about the possibility of Down syndrome, which is an aneuploid. It's caused by aneuploidy. So you can diagnose these by karyotyping. So the figure that we're looking at here, as you should know by now, is a karyotype. And this is a female because there are two X chromosomes. So right there are the sex chromosomes. There is not a Y chromosome. There are two X chromosomes. So that's what makes a female, biologically anyway. And this individual also, you'll notice, and they try to point it out here by changing the color of those chromosomes. There's trisomy 21 here. One extra chromosome. So there are 3 homologous chromosomes here for chromosome 21, and that's trisomy, and it causes Down syndrome. And Down syndrome is named after John Langdon Down, who in 1866 was the first to describe the condition. And this is an instance where having extra genes is a problem, and the production of too many proteins, certain proteins, is a problem, a developmental problem. And those individuals that have Down syndrome, each is an individual, meaning that the effect of the disorder is highly variable. Some individuals will be healthy, while others have a lot of health problems, and some individuals will be more or less mentally affected. So each case is different. So this is an example of aneuploidy involving an autosomal chromosomes, but we can also have aneuploidy involving sex chromosomes, and that's what we're looking at on this slide. These are 2 different forms of disorders that are caused by the wrong number of sex chromosomes. And they're caused by non-disjunction of the sex chromosomes, just like in the autosomes. So if you analyze this karyotype, what you find, and they put a box around it to make it evident, is that this individual has 1 X chromosome, and that's it. It's not 1 X and 1 Y. It's not 2 X chromosomes. It's just 1 X chromosome. So there's a missing chromosome here. Monosomy. Right? This is an example of monosomy. And so this is a female, biologically a female, because she doesn't have a Y chromosome. And remember, the Y chromosome is what turns a male into a male. So basically, the way you want to think about it is we all start out female, and then those of us that have a Y chromosome, that turns us into a male. So this individual has Turner syndrome, which is caused by non-disjunction and monosomy of the sex chromosomes, which was first described by Dr. Henry Turner in 1938, and that's why we call it Turner syndrome. And you can see some of the symptoms by looking at the picture of this girl here. The primary symptoms include being short, with puffy hands and feet, organs are affected, and therefore may malfunction, and individuals may be unhealthy as a result, along with being sterile. In other words, people with Turner syndrome can't reproduce. Reason being that the wrong number of chromosomes somehow interferes with meiosis and gametogenesis. If we analyze the karyotype of this individual, we will note that they have two X chromosomes along with a Y chromosome. So biologically, this is a male because the Y chromosome turns a male into a male, or is what's responsible for causing a male to become a male, develop into a male. But there's an extra X chromosome here due to non-disjunction, So this is an example of trisomy. And this individual has Klinefelter's syndrome, named after Harry Klinefelter, and he described this condition in 1942, so relatively recently. And you can check out these symptoms for yourself from the diagram, but in general, the primary symptoms are general feminization and also sterility. In other words, these individuals cannot reproduce, and again, it's because there's the wrong number of chromosomes, and that interferes with meiosis and gametogenesis. And there are other such disorders, and if you're curious about those, you can click. There's a link here, but it's not going to work in the video, but it does on the note guide. And that's really it for the human heredity part of this unit. What we're going to get into now is actually biotechnology, even though it's part of Chapter 14. Chapter 13 is supposed to be about biotechnology, but like I said, biotechnology is pretty much based on human heredity, and so we need to investigate both at the same time, investigating both together. So the first biotechnology we'll be looking at is how it is that DNA fingerprinting is done. And this is also an example of how genetic testing can be done. And we're going to create a crime scene scenario here to explain how DNA fingerprinting works. And it's based on the fact that there are differences between the DNA of individuals. So what we're looking at here is a diagram representing DNA. So you can picture this as a double helix. Even though it's being shown here as a bar, you can picture it as a double helix. And the first thing you need to do DNA fingerprinting is to have DNA samples. And so that's, again, what we're looking at here, samples from two individuals. And you can obtain samples of DNA through DNA extraction from cells. So anytime you can get a sample of cells, you can extract DNA from those cells and amplify that DNA through a process called polymerase chain reaction, PCR, which is another biotechnology we'll be looking at later. And then you'll have enough DNA to be able to analyze, do DNA fingerprinting and or genetic testing. But in this scenario, we're going to imagine there was a crime. So let's say there was a shooting. Somebody was murdered. And at the crime scene, there was a cigarette butt lying on the ground where the shooter was. So along with some shells from the bullets, there was a cigarette butt. And from that cigarette butt, there are cells from the mouth of the person that did the shooting. So they have cells from the suspect. Or actually cells from the perpetrator, the person that committed the crime, and then these two DNA samples are representing two possible suspects. So we'll consider this to be suspect 1 and this to be suspect 2. And just to remind you here, what genes are, notice that there are genes in these DNA, what are representing these DNA sequences, and genes are sequences of DNA that code for polypeptides or proteins. And the idea here is that gene A and gene B and gene C, as you can see in the diagram, there's no difference between them, between these two individuals. And DNA fingerprinting really doesn't rely on differences in genes. There can be differences in genes. In other words, for gene A here, these two individuals might have different alleles for those genes. And you could find that out doing DNA sequencing, but the procedure we're looking at here, the DNA fingerprinting, relies not on differences between genes, but differences between non-coding regions of the DNA that are labeled here repeats. More specifically, they're referred to as short tandem repeats, STRs. And they're short sequences of DNA, 2 to 5 base pairs long, that are repeated numerous times. So like in this case, GATA, GATA, GATA. So it's 4 repeats of GATA, GATA, right? And that would be possibly what we have here, GATA, 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 GATA. Oops, I just got carried away and I said it 5 times, but you get the idea. So notice that the number of repeats are different between individuals, and that's what DNA fingerprinting relies on, or is based on. The difference in the number of repeats between individuals is what we call polymorphic, right? Polymorphic, poly, many, morphic, shape or form. So the form of repeats, or the number of repeats, is different takes on a different form between individuals. So there are many forms between individuals. So what DNA fingerprinting does is identify the difference in the number of STRs between individuals, between the number of short tandem repeats between individuals. And we'll get into how on the next couple slides, but I just need to point out here that these differences can also be used as what we call genetic markers in doing genetic testing. So say, for example, gene B is a gene that is involved in certain kinds of cancer. And we want to see if the individual that has the recessive allele that is prone to causing cancer, having a certain number of repeats next to that gene can indicate a certain allele for that gene. So maybe 4 repeats indicates that gene B here is the recessive allele, whereas 6 repeats indicates that gene B is the dominant allele. So the number of repeats can indicate a particular allele of a particular gene. So what's the procedure for doing DNA fingerprinting and comparing the number of repeats between these two individuals? Well, here we have suspect 1 and suspect 2 again. And you'll notice that we're using a way to cut up the DNA. And the DNA is being cut up in a very specific way. And we use these things called restriction enzymes to do that. And restriction enzymes were discovered in bacteria. Restriction enzymes act as an immune system in bacterial cells. So like, for example, you remember bacteriophages are viruses that infect bacteria. The restriction enzymes in the bacteria will recognize the sequence of the genome of that virus, in other words, the DNA sequence of the virus or the RNA sequence of the virus. And when that DNA gets into the bacterial cell, the restriction enzymes will recognize it and cut it up. So we were able to use the immune system of bacteria as a tool to cut DNA. These are also known as endonucleases. Nuclease because they cut nucleic acids. Right? They're an enzyme that cuts nucleic acids, but we also call them restriction enzymes. All right? So in this case, we're using a very specific restriction enzyme called EcoR1, and the DNA, EcoR1 cuts at a very specific sequence, a recognition sequence or a restriction sequence at a restriction site, as we say, and it's always going to cut at that specific sequence, and not only at that specific sequence, but between two specific nucleotides of that specific sequence. And all of these bluish ovals here are representing EcoR1. In other words, we're using the same restriction enzyme, to cut not only the DNA of suspect 1, but also to cut the DNA of suspect 2. So EcoR1 is a restriction enzyme. It cuts DNA only where it finds specific recognition sequences and at a specific recognition site. And just to let you know where EcoR1 comes from, Echo is short for E. coli, and R1 because it was the first restriction enzyme identified in E. coli, and actually it was the first restriction enzyme ever discovered. And more specifically, this enzyme cuts between the G and the A of the sequence GAATTC. In other words, this restriction site right here, that's where the enzyme found the sequence GAATTC, and it then cut between the G and the A of that sequence. And that's the only place it will cut. These enzymes are very specific to the recognition sequence that they will cut, and then between the two nucleotides that they cut. And these sequences are palindromic, meaning that, if you think of this as being a double helix, it's two nucleotides. There is GAATTC maybe up on on one strand, and then on the other strand there would be CTTAAG, which is the palindrome of GAATTC. So that's how these recognition sequences tend to be. They tend to be palindromic. And that's important because then both strands get cut. The restriction enzyme will find the same sequence, even though one of them is backwards to the other. It will still cut between the G and the A of both sequences on both strands of the DNA, both polynucleotides. So once cut by restriction enzymes, we end up with a number of fragments, and those fragments vary in their length, but it's only the repeating regions that vary in their length. The genes don't vary in their length. Gene A, gene B, and gene C here are the same length between these two suspects, between these two individuals. So again, you can't use the genes to compare in this method, but you can use the repeating regions to compare. So what we have here are restriction fragment length polymorphisms. These are restriction fragments. Once we cut the DNA up, we have restriction fragments because they were produced by restriction enzymes, and their length differs between individuals, length polymorphisms, right? So these are known as restriction fragment length polymorphisms. RFLPs for short, right? RFLPs. That's how we actually refer to that acronym. But it stands for restriction fragment length polymorphism, and it refers to the difference in length of restriction fragments between individuals. That's the basis for DNA fingerprinting. That's what makes DNA fingerprinting work and show differences between individuals. So now that we have the DNA cut up between these two suspects, and we know that there are differences in the length of the fragments, how do we find those differences? How do we measure those differences between the length of the fragments? Well, we go on to analyze the DNA using a process known as gel electrophoresis. Gel electrophoresis. So it tells you right here that it involves a gel, like Jell-O is a gel, and electricity. Some kind of process that uses gel and electricity. So this apparatus is showing the setup for the basic setup for doing gel electrophoresis, this diagram. And gel electrophoresis is the process that separates DNA fragments or proteins. I just want to point out here that this same process can be used to analyze proteins according to size and charge. So this is what we're using to separate out the fragments that were produced using restriction enzymes to find these RFLPs, restriction fragment length polymorphisms. So what we use is an agarose gel, and agarose is the same agar that you guys used when you did the lab where we grew bacteria. The agar was in the bottom of the petri dish, and that's how we did cell culture. We grew the bacteria on the agar. Well, this is the same kind of stuff, and it's derived from algae, but this is a highly purified version of it that sets up a matrix that the DNA can move through. The different DNA fragments can move through. Because it's a semi-solid, it's very porous, and the DNA fragments will be able to move through it. So it starts by placing the samples, the restriction enzyme digested DNA samples, into the sample wells at the cathode end of the gel. So here we have the sample wells, each of these little slits in the gel, and this is the cathode, the negative electrode. So you'll notice we're setting up an electrical field across the gel. And sample 1 is in this lane, as we say. These are lanes. Each sample well represents a lane on the gel. And sample 2 is in this well. And the other wells are just empty in this example. And the whole premise here is that DNA, which is what we're putting into these sample wells, is negatively charged. DNA has a negative charge. Therefore, it migrates towards the anode, the positively charged electrode, because opposites attract, right? So what moves the DNA through the gel is its attraction to the anode. So when we put an electrical field across this gel, that causes the DNA to move through the gel. And that's significant here because the smaller fragments are going to move faster than the larger fragments. So these two bands here that we see represent fragments, and they represent fragments that are all the same size, that are larger. I'm sorry, smaller. And when I say they're all the same size, I don't mean that these two bands, the fragments in these two bands are not the same size, because you'll notice that one is lagging behind the other. This one moved the farthest, therefore it has the smallest fragments, the smallest DNA fragments, and then this one moved a little bit less far, therefore it has somewhat larger DNA fragments. And it's the same for these. This band here represents DNA fragments that are larger still, and then the largest are these DNA fragments. The band here that represents the largest DNA fragments. But within each band represents the same size fragments. So what this process does is it separates the fragments out into groups, which produces bands on the gel. And you might recognize this as being similar to chromatography, the paper chromatography that we did with leaf pigments and the ink pigments that we separated out in the lab. The same thing's going on, just like the ink moved up the paper and separated out the pigments from the ink, or the leaf juice moved up the paper and separated out the pigments in a leaf. The same thing's going on here. The only difference is we're not using paper, we're using gel instead, agarose gel, and we're not relying on capillary action for the movement of the pigments through the paper. We're relying on an electrical field to move the DNA through the gel. But it's really the same thing. Now you have to realize that what we're looking at here is a diagram. And in reality, when we do this, you can't see the DNA in the gel. When you run the gel, when you turn on the power and the DNA moves through the gel, you can't see the DNA in the gel. You have to do something to visualize the DNA. So the DNA is not visible in the gel, so you either stain it, you use a DNA stain that stains only the DNA and not the gel, or you use a radioactive probe that will actually bind to the DNA to visualize it. And in this way, you can use very specific radioactive probes that will bind to very specific sequences in the DNA, and therefore you can use that to identify specific genes, specific sequences, if you're doing genetic testing. So again, this same procedure can be used for either DNA fingerprinting or genetic testing. In this scenario, what we're looking at here is an actual results and a stained gel. So this gel's been stained, and that's why it's a blue stain. So the gel itself takes on a bluish tint, but the DNA is what gets stained very dark because the stain actually binds to the DNA. And it's this pattern of bands on the gel that is the DNA fingerprint. In other words, this is the DNA fingerprint of suspect 1, and this is the DNA fingerprint of suspect 2. And then we also have the evidence on this gel, the evidence being the DNA from the cells that we found on the cigarette butt. So we're making a comparison here, trying to find out who did it, who committed the crime. Was it suspect 1 or suspect 2? And I think you can very clearly see that it was suspect 2 that matches up here in the DNA fingerprint. The DNA fingerprint from the evidence matches up with suspect 2's DNA fingerprint. And you'll notice that the bands aren't exactly the same. The important thing is that they're in the same position. We have 2 bands here, 2 bands here, and they're kind of muddled together, but there are 2 bands, and then we have 1 band here and 1 band here, and they're at exactly, these were run together on the same gel at the same time, and they're exactly in the same position along the gel, which indicates that they are exactly the same fragment length. So that's how DNA fingerprinting is done. And just to summarize, we take DNA samples, we cut them with restriction enzymes, we place them onto a gel, we run them on the gel, which separates out the different fragments that resulted from restriction enzyme digestion or cutting up. And that pattern, the pattern of the bands on the gel is what indicates the DNA fingerprint. So that's one bit of biotechnology that's been developed. And here we're looking at gene therapy. The promise, because at this point it really is a promise, it hasn't been perfected. It's just now being used to cure certain things. But the promise here is the ability to replace a bad gene with a good gene. In gene therapy, the whole idea is that you can take an individual that has a genetic disorder, and that genetic disorder is being caused by a mutated gene, a bad gene. And the promise of gene therapy is that you could actually replace the bad gene with the good gene in the cells that it needs to be replaced in the individual. And if there's a bad gene, that means there's a bad protein. So if you replace the bad gene with a good gene, that means you're also correcting the missing or non-functioning protein that the gene codes for. The example we're looking at here is a possible treatment to cure sickle cell anemia, because it involves the bone marrow, and the bone marrow is where blood cells are manufactured. That's where the stem cells that produce blood cells come from the bone marrow. So that's what we're looking at in this scenario. Just generally, we need to define the term vector, and I believe we've discussed vector before in class, but a vector is anything that carries or transports or delivers something. So, for example, the vector of malaria is the Anopheles mosquito. The vector of sleeping sickness is the tsetse fly, and that's when we were discussing it before in class. So in this case, we're using a virus as a vector to carry the good gene into the cells, right? That's what viruses do. They insert their genes into cells and and their genes then take over the cells. Well, in this case, we can genetically engineer a virus. So we're going to use the virus as a vector and genetically engineer it to have a human gene, a good gene, that's going to replace the bad gene in the patient. And we can do this through genetic engineering, which is another biotechnology that we'll be looking at later. So this is kind of a combination of a couple different biotechnologies in gene therapy here. But viruses are thought to be, and I say thought to be because, again, these procedures really haven't been perfected, and viruses have been shown to do this fairly well. But there have been some setbacks too, some situations where they've tried this and it's actually killed the patient. So that has set back the technology quite a ways, number of years, but it's coming back now. But viruses infect specific cells, if you'll recall. They're like smart bombs. They only go after specific cells in specific species, and they insert their genes into those cells, like I already mentioned. So this is a bone marrow stem cell, and so the idea would be to take the bone marrow stem cells out of the patient, use the viruses to infect those cells outside of the patient, and then place those cells back into the patient. So viral infection and gene insertion. So there's the virus, the genetically engineered virus. It's infecting the cell and inserting its genes into the genome of the cell. And then those cells can be proliferated. In other words, in cell culture, they will multiply. So you only have to do this to a few cells and then just allow them to multiply. And then you put them back in the patient where they will continue to multiply. They will continue to divide by mitosis. And therefore, they all will be genetically identical to each other. And then they will take on the role of producing blood cells within that individual. And that will cure, in this case, sickle cell anemia. But the same procedure could be used to cure a variety of different genetic disorders. And if you recall, the gene that's bad in sickle cell anemia is the beta hemoglobin gene. Remember, there's an alpha hemoglobin subunit and a beta hemoglobin subunits. And it's the beta hemoglobin subunit or protein that that is affected, or polypeptide that is affected in the case of sickle cell anemia. So that's the viral method, but there's also what has come along very recently is CRISPR. You may have heard of CRISPR, which is a brand new biotechnology that is also based on the restriction enzymes that we found in bacteria. So CRISPR is actually part of the immune system of bacteria, just like restriction enzymes are part of the immune system of bacteria. And it's referred to as the CRISPR-Cas9 system. And again, it normally functions as a prokaryotic immune system, but we've been able to harness that as a tool. And it's a tool to cut and edit DNA at very specific sequences. Just like restriction enzymes will look for that recognition sequence and only cut between two specific nucleotides of that recognition sequence. CRISPR is the same way. It will find and cut DNA, and we can actually use it to edit the DNA, to change a bad gene to a good gene, to change a mutated gene into a normal gene. So at this point, you may be thinking to yourself, well, what the heck does CRISPR stand for? It stands for Clustered Regularly Interspaced Short Palindromic Repeats. That's a mouthful. You wouldn't want to say that all the time, so that's why we say CRISPR instead. But you may recognize here that we're talking about palindromic repeats, just like we were when we were looking at restriction enzymes and how restriction enzymes work. And you'll notice in this diagram, the gene therapy is comparing two different possible methods, one being in vivo and the other being ex vivo. In vivo means inside an organism. So in other words, we would treat the cells while they were still in the individual. So we would treat the whole individual, basically, whereas ex vivo means outside an organism. And this would be similar to what we were looking at on the previous slide, where their cells would be removed from the patient, then they would be genetically engineered, and then they would multiply and then be placed back into the patient. And if you read these, you'll see there's more going on here. Not only will they be multiplied, but they'd be tested for safety. So what they're suggesting here is that they would be injected into mice before injecting them into the patient, just to make sure that there's not an adverse effect in the mice, that it was safe to put those cells back into the patient. And you'll notice we're looking at bone marrow in this part of the diagram again, so this would be, again, maybe the cure for sickle cell anemia, the beta hemoglobin gene. So in comparison here, the in vivo approach would be to place CRISPR into circulation. And it's explaining here that it would be packaged in a particular way to deliver it into the body, but it would be injected into the patient or into the patient's organ or whatever needs to, wherever the genetic engineering needs to take place in specific tissues. But either way, it would go into circulation and it would edit the gene no matter where it's found in all the cells of the body, potentially. And then on the other hand, by removing the cells and genetically engineering the cells outside of the body, only the target gene in the target cells will be edited. And, in the case of in vivo, it's possible that other genes might be affected, accidentally edited, and that can even lead to things like cancer or cause other problems. So what I'm trying to get across to you here is that it looks like the ex vivo approach is a lot safer than the in vivo approach, because its intended cells are less likely to be affected. Unintended cells are less likely to be affected. But CRISPR technology has a huge potential for doing a lot of different things in biology. And it's a huge biotechnology that's continuing to be developed, and they have different kind of CRISPR systems other than Cas9. The Cas9 system, for example, they do all kinds of different things. So it's a real up-and-coming biotechnology. But along with this technology come a lot of ethical considerations. For example, there was, you may have heard in the news relatively recently, there was a Chinese scientist who genetically engineered some girls to not be susceptible to HIV, but he had no authorization to go ahead and do that. Therefore, the Chinese government put him in jail. So these things need to be tightly regulated because they're powerful things. The greatest concern, if we go back to the in vivo example, the cells that might be edited by CRISPR, if they are simply injected into the body and therefore there's the potential to edit all the cells of the body, those cells might include the germ line, what we call the germ line, which are the cells that produce gametes. So spermatocytes, oocytes might get edited, and therefore the sperm that results from those spermatocytes or oocytes would be passed on to the offspring of the individual and their children and their children. In other words, we'd be altering the germline of the human race, which is an extremely powerful thing to think about, and gives us the ability to take charge of our own evolution, really. And ex vivo, we can do that intentionally. We can do that with the cells, the germline, the cells that become gametes, but we can also do it with an already fertilized embryo or zygote. We can use CRISPR to genetically engineer that zygote or that embryo, and then it will develop into a new individual that can pass on that genetic editing to its offspring. So this kind of technology brings up a lot of questions like, just because we can, should we? And who regulates this kind of thing? Those are the questions that we all have to deal with in the future.